Okay, now how can we move from this kind of laboratory setups to real world environments? Okay, so this is that, as I mentioned repeatedly, my main interest is in using synthetic biology, not so much for medical applications and agricultural applications, but for environmental applications. And one typical environmental scenario that we all know that is not desirable is this with plastics and so on. And you know what happens here as a different situation in respect to what you have in a bioreactor is that if you want bacteria to act under these circumstances, there's evidently one thing that immediately brings your attention. What do you think would be the big difference between having bacteria acting here or here versus having bacteria working in a bioreactor? What immediately you would say is different? Well, that's one thing, but more things that to me are immediately okay. That's also an interesting point. More possibilities. Okay, well, to me, the big difference is that bacteria that are in a bioreactor have plenty of water around. You know, they are saturated with water, whereas the bacteria that are working under these circumstances work under water subsaturation. It doesn't mean that they are dry, but they are subsaturated. So that means that they don't have as much water as they have when they are working in a bioreactor. Okay? So obviously things get a bit more complicated when you want to apply this type of bacteria to deal with a soil that is also dry or semi-dry, that is also contaminated. And it happens that big parts of the world in general are quite hot and they tend to form dry lands and so on. So the idea in this case is how to make environmental bacteria to work under subsaturation conditions close to desiccation. Okay, so many bacteria, and in particular Pseudomonas putidum, are very sensitive to desiccation. Okay, so if that's the case, then forget it. We cannot use it for environmental applications because immediately bacteria will die. However, there are tricks to improve the situation. And one of the tricks is to look at other biological systems that resist desiccation very well. And one of the most famous of these systems are the tardigrades. So tardigrades are animals that are microscopic and that have evolved to tolerate super high levels of dryness and desiccation. So these animals are famous because they can go into the desiccation stage and then stay under these conditions for years and years and years and years, and then go back to life in case that they are exposed to water. So apparently, one of the reasons why these animals are so resistant to desiccation is because they produce some proteins that are intrinsically disordered. So that means that they don't have a special structure, they have a kind of different structure that is disordered, and they are important to survive desiccation. So what we did at some point was to make a small catalog of these proteins here, and then it comes from different types of tardigrades. They can be found in the literature. So we identified the genes. The genes were sent out for synthesis. You have now many companies that can do that for you. And then each of the genes was cloned in a plasmid vector of the type called SIBA. You will hear more about that later. But this is a plasmid vector for cloning or whatever, where we have the intrinsically disordered protein, each of this type, under the control of an inducible promoter by benzoate. Okay, so this is a typical construct, synthetic bioconstruct. We have the biosensor model, the inducible model, Schendel-Garno, and then the gene of interest. In this case, the gene encoding a disordered protein that, in the context of these tardigrades, produce some resistance to desiccation. Okay, so and then we made some experiments, and this is what we got. So we expose the cells to low water contents in the atmosphere, and then what you observe is that depending on the different genes that you have here, then you can have up to 100 times more resistance to desiccation than the natural, say, situation. Okay, so this is one of the cases where, by using these tricks of genetic engineering, so you can add. You remember this idea of cyborgization. So cyborgization was both about removing things that you have, but also to knock in things that can be useful for a specific application. Okay, so the last screw turn that I will discuss today on what can you do with a naked bacterium as a chassis for different applications is this idea of engineering the attachment of given bacteria to a specific target surfaces. In the case of plastics, it's very, well, you may think, well, you may have a bacterium in the laboratory that produces some plastic degrading enzymes, for instance, and in some cases you can put them on the surface of the bacteria. That has been done, in fact, but then you want the bacteria to stick to plastic surfaces, not to just hang around. Then, in that case, you have to kind of think on ways through which you produce a type of tropism of the bacterium against a surface of choice. So that involves like three questions. One of them to have a specialized chassis. In our case, that's not a problem because we have this Pseudomonas putida naked strain. You need to have a surface display system for displaying the protein of interest on the surface and something that can be encoded genetically. And also you need the adhesins proper. Okay, and the adhesins proper, as you can imagine, are antibodies. And you have the target, and the target can be another bacterium, as I showed you before, or it can be a molecular motif that is displayed on the surface. Okay, let me just mention on passing different displaying systems that we have domesticated for our own use, that go as follows. You know that many bacteria, in particular pathogenic bacteria, they have ways to release things into the environment to damage the, say, the victim, to damage the host tissue, and there are right now up to, I think, like something like 10 different systems for secretion of proteins in the external medium. And under natural circumstances, these, the proteins that are secreted or that are put on the surface are proteases, adhesins, lipases, esterases, and so on. But in our case, what we have been doing has been to concentrate in this type of proteins that are called autotransporters. So autotransporters are outer membrane proteins that naturally are composed of two domains. One domain is inserted in the outer membrane, and then it has like a kind of ring through which a protein is exposed on the surface and also attached to the surface because of this anchor that is a beta barrel with the nomenclature of structural biologists. So there are various types of various variants of this general scheme, and we have used various of them. One of them is based on the IgA protease that comes from Neisseria gonorrhea, that is a bad pathogen, and then other two come from E. coli, but not our domesticated E. coli, but from an E. coli that is pathogenic, and the intimins are also coming from another E. coli that are pathogenic. So the E. coli strains that we use in the laboratory are completely domesticated. They are not dangerous at all. But E. coli in itself is a bacterium that has many, many pathogenic strains in the group. And the worst thing that may happen to you is that you go to a hospital and you get a pathogenic E. coli. I mean, this is something that can be very bad, or you can have an intestinal infection, well, you name it. But it's just to mention that we have recruited this display system from pathogens, and we have, say, recycled its activity not to display something bad, but to display something good. And that good thing is these antibodies that I told you before. Okay, so someone in my laboratory called Sofia made this project, and the question is whether we can stick naked cells to a surface complex with an adhesion partner. Okay, so we have this naked cell, we have a surface where we have a kind of bait, and then can we bring about a specific adhesion of these naked cells to the surface Well, this is something that we try, and at least theoretically, it would be possible if we make bacteria, Pseudomonas putida, to display on the surface an antibody that interacts with the antigen that is coating a given surface, okay? So just to get a proof of concept, okay, I have to make a little parenthesis. So proof of concept is developing an experimental system that is not the reality, but it captures the essence of the real situation that you want to address, okay? So in this case, I mean, the real situation that we want to address is to see whether we can attach Pseudomonas putida to plastic. But you know, this is too much into practice, too much into application. We have to go a few steps back and then make a super simple model to see whether, number one, we can attach bacteria to a surface, period. Not to a plastic surface, but to a surface that is characterized by some molecular motif. So just to develop this proof of concept, then we took an antibody that responded to a protein that is called fibrinogen, and then we had a surface that was coated with fibrinogen, okay? So the prediction is that if everything works, we have bacteria here that will not recognize anything excepting a surface that is coated with the fibrinogen. And this is the experiment. So, well, there's some technical details here. So the way you measure, well, there are various ways to monitor whether bacteria are sticking to surfaces. Some of them are sophisticated, others are very simple. The simple is the microscopy, right? So this is a surface that is not coated with, well, just the contrary. This is coated with fibrinogen, but the bacterium is not displaying the corresponding antibody. Then you see when this bacteria goes through this surface, the surface is ignored. So the bacteria passes by and ignores the surfaces. In this case, however, we have this bacterium that displays on the surface a nanobody, antibody that will recognize this antigen here, and then you see that the cells form a nice monolayer. I insist that this is a monolayer, it's not a biofilm, you know, because in this case, bacteria do not interact with each other, they just interact with the surface, and therefore, the moment the one bacterium grows, it will go back to stick as a monolayer to the surface. It will not go out or anything. Okay, so that's something that is nice. So this is visual, but then you can monitor, you can count the number of bacteria and the biomass that gets attached to the surface by using a microbalance. Okay, so it's a technology that is a bit complicated, but basically the difference between this line here and this line here is the microbalance, the weight of the bacteria that sticks to the surface as compared to the control with no attachment there. So obviously with this parameter you can make many calculations. How is the weight of one bacterium, how many bacteria you have, the time of residence. I mean, you can make many parameters here. But the essence of what I'm telling you is that by making this trick, you can have bacteria really interacting with a surface in a very specific fashion. Okay, so just before we go into our, second part or the last part of today's classes, let me just recap a little bit the situation of the different groups. So we have the team number one, the Bomita, that is not here today. We have Abdul, that is here, Ashna, I'm sorry that I still have to need time to remember your names. Tejas is not here today. Okay, so this group is presenting today? You are presenting, yeah, okay, very good. Did you load your presentation?
Oh, yes.
Okay, excellent. Then number two have Sanchin. Oh, yeah, you are Sanchin. Puja, not here today?
She was here, she actually left, she has a class.
Yeah, okay. Pranati, yeah, and Pashinavi. Okay, very good. So who will be presenting? Excellent, very good. Then we have Adita, yeah, is there. Yeah, sorry. Adit, very good. Abhijan, excellent. And Rajesh. Okay, so it says that in every group there's a defection, that's interesting. And then number four we have Meg, okay, very good. Mutu, and Sunko. Okay, one of you told me that you were the one that told me that you may not make it. You have to leave. So what do you want? You prefer to talk tomorrow or you prefer to talk now?
I don't know if I'll be able to finish it in 15 minutes.
So. Okay, so why don't we do one thing? So we leave your talk for tomorrow, okay? Very good. Okay, so this is the, okay, very good. So we have like representatives of all groups. That's something very well. By the way, did the arrangement work well? Did you have your discussions and everything? Or there were some cheaters? You know what cheaters are? So cheaters, you know, there's this, in microbial ecology, there's this theory about cheaters, you know. So cheaters are those members of a community that benefit from having some common good, but they don't contribute to the common good, okay? So it's like, you know, you have a party, a potluck party, everyone has to bring something, but there are some guys that go there, eat everything, but they don't bring anything, right? So that's the, so well, that's sometimes unavoidable in a cooperative. There are even mathematical theories to explain these things, how much cheaters a community can sustain before it collapses. You know, there are interesting stories about that. Well, anyway, so well, I'm happy that at least today most of the participants, most of the teams have representatives.
So, hi everyone, I'm going to be doing a small analysis on this paper. So, as a small introduction, this paper deals with GrowEL, which is an essential like molecular chaperone that has a lot of in vitro applications that could be very useful. However, the main issue with GrowEL is that it is very difficult to the main issue with GrowEL is that it doesn't undergo substantial evolution, and hence there's a need to create like an orthogonal start over. Sorry.
Don't worry, you are between friends, among friends, so don't be nervous.
Of GrowEL is considered difficult due to its complex structure. So this paper essentially describes the first successfully individualized GrowEL genes, which could possibly serve as like a strong basis for the evolution of GrowEL. So this has been done using by inserting functional peptide tags into permissive sites that are identified by transposon mutagenesis. So this allows for the genes to be transposon mutagenesis allows for the genes to be transferred to a host organism's chromosome by and it interrupts or modifies the function of the extant genes on the chromosome to cause mutations. So there are two main prerequisites for the diversion of important cellular functions, and they're listed over there. So the first one is that the relevant genes must be duplicated so that there is a liberated copy of the gene that can evolve separately from the original one. And second, the gene products must be separated from the remaining gene so that the evolution takes place in an orthogonal manner. So this is especially relevant when dealing with homomultimeric complexes. And GrowEL is an example of a homomultimeric complex, and hence this is especially relevant. So these prerequisites can ultimately result in the orthogonalization of the old and the new functions. Coming to GrowEL, so as mentioned earlier, it is an essential complex that is homomultimeric and has multiple functions. And it's an obligate protein folding machinery for around 85 different proteins, and it interacts with around 250 proteins. So it has a very essential function in the cell. So there are two specialized nanocompartments in its structure, and there's a co-chaperone called GrowES, which acts as a cap for the two chambers. And so the structure of GrowEL has two levels of complexity. One being that seven of the Groyer monomers form a ring, and the second being that the two single rings form a functional double ring. So, coming to the functioning of Groyer, polypeptides that are to be prevented from aggregation are directed into one of the two cavities of the structure, and they undergo defolding inside the chamber, which is capped by Groyers. And when the refolding happens, the Groyer undergoes a strong conformational change driven by ATP, and the refolded protein is released from the structure by allosteric binding of Rho-ES. So to implement engineering strategies for Groyer, a method for generating biochemically distinguishable gene products for like different Groyer genes would be pretty important. So in this paper, as I mentioned earlier, they've utilized the insertion of a peptide into a permissive site, which they've identified by transposon mutagenesis. Coming to the results, so the first subheading was in vivo selection of functional Groyer variants containing protease cleavage sites. So essentially, the mini TN5 transposon system was utilized to find permissive sites, and two of them were found after screening. One after residue I301 and one after residue D523. Also, two Groyer variants containing the PPD cleavage sites together with the remaining amino acid residues were like analyzed as a whole. So this is the visual representation of the Groyer structure, and we can see the I301 residues marked and the D523 ones marked on the inside of the chamber. So one of the aspects that was explored in the paper was ensuring that the inserted peptide, like the results obtained due to the analyses were not just exclusive to the inserted peptide, and for this, they replace the PPD recognition site by a TED protease, and this led to two additional variants, which were also considered in the further analysis. And this was one of the main aspects of the paper that I appreciated, because I felt like this made the results obtained in this paper more robust, more accurate and thorough. Coming to the influence of the peptide insertions on the overall performance of Groyer as a chaperone in vitro and in vivo. Biochemical analyses were run for the 4 Groyer variants, and similar modalities were coming to the results. We saw similar modalities on like polyacrylamide gel and comparable ATPase activity as shown in the graph. Also, the variants displayed intact but slightly substrate-specific refolding of the molecular chaperone synthase. When we looked at the functionalization and individualization of Groyer through the peptide insertions, it was observed that the I301 site was reasonably tolerant for insertions of segments. Individualization of Royal monomers allowed the straightforward removal of the first obstacle to Royal engineering, but it still leaves the potential problem of monomers from different genes assembling into the same complex and interfering with its function. So to analyze this, individualized Royal monomers were used to track the Royal complex composition in the presence of different monomers. And it was found when pull-down assays were conducted, it was found that the ring structures were strongly biased. Also, the last point, the rings seem to be recruited differently. But I wasn't very clear on this portion. I wasn't able to understand the paper. I want to just clarify about that. But I think that's about it for the results.
Okay, thank you. Okay, so number one, did you like the paper? Right. Okay, so what is the main message that you get out of the paper? So what will you remember about this paper? Apart of some difficulties to understand the techniques. I mean, what do you think this paper delivers in terms of knowledge or in terms of ideas? What do you think this paper is good for?
To utilize orthogonality to create more to basically utilize this Groyan as a possible basically utilize Groyan as like a possible tool for the analysis and research.
Any criticism or any appraisal?
Two additional variants were considered after the PPV protease. I think PEV PEV protease. So I thought that this was a good aspect to consider since it makes sure that the results were not simply because of the protease that was used, and it kept like the results more independent of that, and hence it makes the results in the paper more accurate and thorough.
Okay, very good. Any questions or comments? No? Okay, well, thank you. Thank you. Okay, next presenter.
Hi, everybody. So today I will be explaining what I understood of a paper titled Engineering Stability in Gene Networks by Autoregulation. So most biological systems have a tendency to maintain constant internal chemical and physical conditions, and this is called homeostasis. And so in genetic and biochemical networks associated with homeostasis, perturbations in transcription, translation, and protein degradation rates must be withstood. And it is proposed that auto-regulatory negative feedback loops in genetic networks provide stability, and stability is another term associated with homeostasis. And as so in biological networks, in biochemical networks and genetic networks, when the concentrations of the various network components remains the same or similar in a steady state, we call it stability. And in this paper, I noticed that they compared stability, homogeneity, and variability amongst in auto-regulatory networks and unregulated networks. So So negative feedback loops. So it's constructed using a transcriptional repressor and a regulator. So the repressor goes and binds the regulator and prevents the transcription process. So it's the general principle of the negative feedback loop. And so for constructing an autoregulatory system, they have used the tetracycline repressor, which is fused with the EGFP, which is enhanced green fluorescent protein, and that is placed downstream of the lambda promoter, which has two of the tetracycline operators. And so we have two controls for this, one with the TET EGFP repressor mutated, and another one with the TET operators being replaced by the LACI operators. So in these cases, the binding between the repressor and the promoter will not be strong, and hence it's not auto-regulated. So we have B and C are examples of unregulated systems. And
in this case, what is the difference between this case and the case A? No, B is clear, but what is the difference between A and C? Okay, got it, yeah. All right, good.
so they have used differential equation models to understand the stability. And I was not able to understand the differential equations per se, but these were the results that were mentioned in the paper. So the equation of our interest is SR is S auto by S unregulated. So that's the ratio of the stability of the auto-regulated and the unregulated systems. And we can notice from the equations that the stability of the auto-regulated system decreases with the binding of repressors and with lower transcription rates. So that is what we can see in the graph. So as the KR value increases, the SR ratio also increases. And we can see that as the half-life increases, again, the stability ratio also increases. Oh, another thing that we can deduce from this is that S auto is greater than S unregulated. I think the minus K DG formula was also supposed to be there. And so another concept mentioned in the paper is variability. So we are trying to understand the variations in the intensity of the fluorescence that is seen in the auto-regulated and the unregulated systems. So the auto-regulatory system has low steady state levels, but high levels of homogeneity. And so even when we use different plasmids with different copy numbers, the variability remains the same. So that is not a factor with respect to the plasmid copy number is not a factor which affects the variability. And we can also see that in the graph. So A is the auto-regulatory system, B, C, D are unregulated systems, and the variability of A is much lesser than B, C, D. And so to study this better, they tried using inducers, particularly inducer ATC, which is anhydrotetracycline, was added. And this is an inducer of the TET operon. And when low concentrations of the inducer was added, the auto-regulatory system was able to work well. But as the concentration of the inducer rised, it transitioned into an unregulated system. And as we saw in cases of B and C, which was with mutating the repressor and with replacing the operator with the LACI operator, I'm sorry, the variability has increased, which again we can see from the graph. This in case of the unregulated system with the TET, the TET operator being replaced with the LACI operator, we tried using IPTG as an inducer to increase the expression of theta GFP. And the fluorescence intensity obtained is very broad as compared to the autoregulatory system. And space like. So as we can see, in case C it's very broad, and in case A, which was autoregulatory, it's narrow. And another thing that was noticed was that with adding IPTG to the system, increased the variance of the network. Even with time, the variance did not decrease, which was another. Homogeneity is so another thing that I'm trying to say about homogeneity is that it's not just a consequence of the low steady state levels of TET EGFP. So to show this, the fluorescence levels of the autoregulatory and the unregulated systems was measured, and the values were equal. The average fluorescence levels appear to be equal, but the variability of the unregulated system was almost 3 times the variability of the autoregulated system. And we can again see that from the graph approximately. Yes. So a couple of things that could be deduced from this was that the stability of the autoregulated system is definitely higher than the stability of the unregulated systems, and variability of the unregulated systems is greater than that of autoregulated systems. And a couple of points mentioned in the paper that I found interesting is that deficiency or excess of the transcription factors can lead to growth and development disorders in the cells. And negative feedback helps in ensuring homogeneous distribution of transcription repressors. And variability can be advantageous to evolution and stability for homeostasis.
Okay, thank you. Are you done? Okay. Okay, just a little formal thing. So every time you give a seminar, I suggest you to finish with a grand finale, don't just stop. I say, well, and in conclusion, this is a wonderful paper, and I'm very happy that you have listened to me. I mean, something that if you just interrupt without any grand finale, then people don't know what to do, right? But okay, well, thank you. It was a wonderful presentation. So, what to me is the virtue and the beauty of this paper is that at the end of the day, everything is around 3 constructs and very simple constructs that each of you can do in 2 weeks, right? The constructs. And then what they have made is this entire conceptual, say, elaboration, that by having this feedback loop, then you kind of make the distribution of in the response much sharper. If you don't have it, then things get spread out. You have a mathematical model and so on. But to me, what is really interesting about this paper is the simplicity. I mean, you may, if you don't know mathematics, I don't know many mathematics, but you may believe that the equation part is the difficult one. You are wrong. I mean, making an equation is very simple. You can do it in days. But making a construct may take years to make, right? So typically, many of these synthetic biology papers involve, 1 week of mathematical modeling and 3 years of making the right constructs, right? However, when you read the paper, it looks like if everything is the mathematical model, and then, like the experiments are like piece of cake, right? So this is something that is quite remarkable. And this paper is considered also one of the foundational papers of synthetic biology. Okay, so that's very good, despite the simplicity as compared to the repressilator, as compared to the bistable switch. Okay, but still, I think this is a very good contribution to show that you can engineer a feedback loop. By doing that, you really change the characteristics of a given system, right? So that's very good. So any criticism or any comments that you?
In the repressilator, I noticed that they hadn't quantized stuff much. They kept using words like strong and weak binding. But here I noticed that they quantized everything including variability and homogeneity and everything, which I found very interesting.
Okay, excellent. Any question or comments? No? Okay, thank you so much again. Okay, and last presentation of the day.
Almost evening, so good evening, everyone. I'll be giving a short review on the review paper, which is on designing VLP or virus-like particle enzyme nanocompartments. So, what this paper aims to, why are we doing this in the first place, is to essentially implement orthogonalization. And the actual heading or like the title of the review paper is Harnessing Physicochemical Properties of Virus Capsids for Designing Enzyme Confined Nanocompartments. So it was written by these absolute legends. So moving on. So what are VLP? So virus-like particles are essentially non-infectious protein cages or capsids, which are in which the viral genetic material or the genome is stored. So in case of actual phages or viruses, these things are infectious and have virulent factors on them on the surfaces. But in case of virus-like particles, we can engineer them to remove the virulence factors, and they provide a really good environment for the encapsulation of enzymes and provide like a kind of nano reactor, so you can facilitate a lot of chemical reactions which require and certain enzyme concentrations, certain pH levels, and also we can control the fluxes of substrates, intermediates, and products across compartments. And also sometimes what happens is when we are trying to use enzymes, the enzymes might be prone to degradation from either thermal degradation or protease degradations. So sequestering the enzymes in such a manner into the virus-like particles helps to prevent such disturbances in the reactions. Virus-like particles are typically comprised of single coat proteins or CPs as they're referred to. These are in contrast to the bacterial microcompartments, which are generally composed of homologous coat proteins, which are uh, yeah just yeah they're in contrast to bacterial microcompartments whose shells are generally composed of multiple homologous proteins. So these can also serve as a simplified model to study bacterial microcompartments, and we can analyze them further to build a better model or do analysis of the bacterial microcompartments. So there is a few ways to implement encapsulation inside virus-like particles. So one of them, like the very primitive, the very initial method was passive encapsulation, which involved the essentially what you see in the diagram. So this is a CCMV or cowpea chlorotic mottle virus. This involves just the dismantling of the capsid at a particular pH, where the capsid coat proteins just destabilize at a certain pH and disassemble, and you introduce them in a solution along with the enzyme of interest, and at another certain pH they just self-assemble to into the initial capsid form. But this time the enzyme is just trapped inside. So this happens at certain pH values, and it can essentially self-assemble. But this is a very slow process, and that's why hence it is passive encapsulation, and it's not facilitated by any of the enzymes or any other molecules. So one of the papers which involved or demonstrated this was Cornelissen. One of the major scientists who was involved in the demonstration of this was Cornelissen, and he demonstrated that horseradish peroxidase was successfully encapsulated into a CCMV virus-like particle, and the enzyme activity did not was not affected significantly because of the encapsulation, and also this also proves that the peroxides which are the substrate for the horseradish peroxidase enzyme, were allowed very free entry and exit from the virus-like particles. So there is another method of encapsulation which is called the electrostatic interaction mediated encapsulation. So this involves the use of sorry, excuse me. This involves the use of genomic RNA, or essentially charged regions can facilitate. So as we saw in passive encapsulation, the encapsulation rate was very slow, which increases in presence of the genomic RNA. So uh, the strong electrostatic force between the code proteins and the RNA provides the initial driving force for the assembly of the whole capsid or virus-like particle. And using this concept, we can engineer charged regions onto a protein or the enzyme of interest which we are trying to encapsulate and facilitate this process of virus-like particle formation. So this is not only done naturally by the RNAs which are present in the viruses, but also can be done with inorganic ions, inorganic molecules, which have negative charges or complementary charges, which can help facilitate the formation of these virus-like particles. So, this scientist named Hilbert, he also showed that genetically engineered mutants of limousine synthase from Aquifex aeolicus having negatively charged internal surface was assembled using GFP-R10. So GFP-R10 has a large arginine, 10 residue arginine tag at the end, which is a positively, like, it resembles a large positive charge, and the AALS stands for the Aquifex aeolicus. Limousine synthase, which has a negative charge at the end of it, and essentially the initial driving force for the assembly of the capsid is provided by the interaction between these two particles, these two molecules. So, yeah. Another method which was implemented was the RNA tagging signal mediated encapsulation. So specific sequences of the genomic RNA were shown to guide the formation of the capsid. So certain sequences which had stem loop structures, for example, they bound to the CPs and formed the capsid. When a protein is bound to ends of certain stem loop structures like these, we can help the proteins of interest get encapsulated easily, and the covalent binding of the stem loop structures with the protein of interest can facilitate encapsulation very easily. So this was also shown by another scientist named Stockley, and he showed that there is a method to encapsulate a cargo protein in an MS2 capsid. This was done in vitro, and essentially there is a small complex called yeah. The genomic RNA of MS2 has a 19 nucleotide translational repressor fragment, which is known to serve as the packaging signal. So the RNA packaging signal, which is mentioned in the heading here, is the 19 nucleotide translational repressor fragment, which once you bind the protein of interest to those 19 nucleotides, it is it essentially is a method to encapsulate the cargo protein into the virus-like particles. So similarly, Finn was also able to show that the enzyme cargoes inside another virus Q beta capsid by another virus Q beta, but he was able to show that these were able to encapsulate in vivo, and the proteins were also co-expressed along with the code proteins of the capsid. So not only we were able to encapsulate proteins which are made separately and then linked to the TR oligonucleotides, but also both prepared at the same time inside the cell, inside the E. coli cell, and then uh, encapsulated using uh, using a tool called aptamers. So aptamers are essentially sequences of RNA, if I'm not wrong, which act like recognition tags. We can use the aptamers to go to certain other the corresponding tags. So here in the diagram, see there's an alpha aptamer. So the red enzyme will bind to essentially the alpha enzyme and the stem loop complex which is found again. So the TR translational repressor fragment binds to the enzyme and we'll get the encapsulation process completed. So another method for the encapsulation was found to be scaffolding protein mediated encapsulation. So scaffolding proteins are another type of proteins which are required for capsid formation in larger viruses. So CPs bind the coat proteins. They bind to these scaffolding proteins, and they scaffolding proteins essentially do the job their name has. So they provide a large scaffold for the capsid to build onto, and it acts as a template essentially. Douglas's lab and the people working there, they were able to harness this, and they were able to use scaffolding protein mediated encapsulation in P22 viruses, which are P22 phages. And P22 virus is a considerably big virus, so the capsid is quite large, and they found that the part of the scaffolding protein which is responsible for the formation of the capsid is essentially boils down to one helix turn helix motif, and they truncated the whole protein without loss of function of the scaffolding protein, which is a great achievement in my view. So this it's a they were able to translationally fuse, if I'm not wrong, translationally fuse the truncated scaffolding protein and the cargo protein to make the complete assembly in vivo. So that's what the diagram shows over there. And they were also able to show in another paper that the encapsulation, this kind of encapsulation can also be used to increase the number of encapsulated particles per capsid. So it gives like a higher concentration of enzymes of interest, which can be put into the capsid. So here in this paper, they demonstrated with alcohol dehydrogenase enzyme, and they showed that there were about 250 alcohol dehydrogenase molecules per P22 capsid, which gives like a really large concentration. It's about 7.6 millimolar, or around 300 mg per ml. So that's almost comparable to the reported value of macromolecular concentration inside cells, which also helps us to which also gives us a tool to analyze how enzymes like this can work in vivo environments. So, for example, if we want, this can be used as a tool for analyzing certain enzymes and their functioning, or the kinetics, enzyme kinetics, or the protein stability, where the concentrations can be maintained at the concentration levels of those inside the cell. So that is another great thing I found about this type of encapsulation. Work has also been done towards the formation of scaffolding protein mediated in vitro virus-like protein formation. This allows more control over the stoichiometric encapsulated proteins by modulating the input ratio of the proteins in vitro. So Yeah, that's probably self-explanatory. They also like conducted experiments with the ADHD, which is the alcohol dehydrogenase enzyme, having a similar um, having co-encapsulated in vivo with the wild type of the scaffolding protein, and this um, increasing the wild type scaffolding protein somehow showed that uh, alcohol dehydrogenase activity reduced in the in the capsules. So this suggested that the alcohol dehydrogenase was suffering from self crowding, or it was not able to regain its activity once encapsulated. But this was again like disproven when the wild type scaffolding protein was selectively removed using guanidine hydrochloride, and there was no change in the alcohol dehydrogenase activity. So yeah, and similar studies for various other proteins can tell us about the effects of crowding and confinement in other enzymes, and we can do much better analysis using these techniques. So yeah. Other non-covalent interactions interaction mediated encapsulations include this another experiment which was carried out in Cornelison's lab, which demonstrated that the positively charged K-coil peptide was genetically fused to the CCMV, which is uh, the virus which I showed you in the first slide. Those code proteins, and the complementary E. coli peptide was introduced to the lipase from Pseudozyma antarctica. Introduction of the coiled coil peptide allowed non-covalent interactions between the cargo and the code proteins before the pH-mediated capsid assembly, resulting in encapsulation of 1 to 4 lipase B molecules per capsid. So this essentially shows that we can increase the encapsulation rates of particular enzymes, considering certain other signals that can be sent, that can be coded for. And there is another under this subheading, there is another class of proteins that was found, which is called encapsulins, and they exhibit icosahedral symmetry, and they have 3 different architectures. And interestingly, all encapsulins have an HK97 like fold, despite sharing little sequence homology with the HK97 capsid. So the code proteins are shown to be similar. Code proteins of the HK97 were shown to be very similar to encapsulins. And yeah, that's about it regarding encapsulants. Yeah, this is another technique which was found to be useful in the formation of a capsid, which is the post-translational ligation methods such as sortase, SpyTag, SpyCatcher, and split in time can be used to form the particles. These methods in MS2 again, which led to the formation of the enzyme being encapsulated in the in vivo using the method of the spy catcher and the spy tag. So as you can see in the diagram, the MS2 was the cargo protein was tagged with the spy catcher, and the MS2 capsid was tagged with the spy tag, and both essentially come together, and the MS2 encapsulates the cargo protein through the spy catcher and the spy tag mechanism. So there's some critical physicochemical properties we need to keep in mind when we think about encapsulations or virus-like particles. These are possible diffusion limitations across capsid shells, electrostatic potential around capsid pores, and pH inside capsids. So the first point is about permeability essentially. Although we have a lot of like X-ray crystallographic data on capsids, we cannot predict the permeability of the capsid shells as they are very dynamic in solution. And so there is this really interesting paper published by again Douglas's laboratory, which showed a way to measure the pore size essentially. So they used dendrimers. Is that right? Yeah. So they used dendrimers which had NADH to they used dendrimers of varying sizes which had NADH in them, and they used coat proteins and they used alcohol dehydrogenases with scaffolding proteins and coat proteins. So they made a complete capside out of encapsulating alcohol dehydrogenases. And to measure the pore sizes, they put essentially different sizes of dendrimers were exposed to the capsid. So whenever the pore size matched the capsid size, pore size matched the dendrimer size, the dendrimer catalyzed the reaction in the alcohol dehydrogenases and it would convert to NAD plus. So that was recorded, and that was essentially that is how they measured the pore sizes. So whenever you found the corresponding dendrimer, you'll get the pore size. Yeah. Another one of the important physicochemical properties was the pH inside capsid. So the high concentration of charged molecules on the inner surface of the capsid can allow a much stronger pH than the bulk environment of the cell. So that is why that is essentially why these can be very necessary in certain enzymatic reactions which are catalyzed only at higher pH or certain lower pH values than the bulk environment of the cell. And these again, Cornelison's lab was able to measure the. So essentially, this is a small problem of measuring the pH inside the capsid. So they found out a way to do it. So that was also one of the very interesting things I found out about this review paper. So they what they did was they sent a negatively charged pH sensitive polymer probe, and that was encapsulated into the capsid, and then the polymer probe essentially gave some kind of indication based on the pH readings. So the pH of the capsid environment was found to be 0.5 units lower than the pH of the surroundings. Yeah. Another important property that affects the stability of the capsids, or essentially the capsid structure or the VLPs, is the presence of genetic material. The viral genome can interact favorably with the capsid or coat proteins to stabilize them, or they can sometimes increase internal osmotic pressure just by increasing the concentration inside the capsids, and this may also destabilize the coat proteins. So this was similar to how the non-interacting genome can destabilize it. Heterologous cargo present inside the capsid can also sometimes destabilize the capsid structure. So the people at Castan and Pablo's lab were able to show the mechanical properties of proteinaceous cargo-loaded P22 with two different morphologies, procapsid and expanded forms, using cryo-electron microscopy and atomic force microscopy. This showed that the cargo increased stiffness of the shell. But there's one interesting mechanism which is behind both procapsid and expanded forms, and it's different for both of them. So the procapsid has the cargo bound to the C terminus of the scaffold protein, Hence, the cargo scaffold protein fusion protein is responsible for the stability in the case of the procapsid. Whereas in the case of expanded forms, it's just the higher osmotic pressure from the environments that causes the extra stability or the stiffness of the shell. Yeah, so this it shows a lot of it shows that a lot of study has been ongoing in the area of the virus capsids, and this shows a lot of interesting avenues that can open up when you're dealing with nanoreactors and places where you can put enzymes which require a certain environment to function at their maximum activity levels. So this is a very interesting topic to like look out for the future.
Okay, thank you.
Yeah.
We think that you were carried away by the enthusiasm for the paper, and then you kind of make a review of the review. Thank you very much. It was great. Questions or comments? Okay, well, I see you so enthusiastic about the technology that, you know, I wonder whether you have some downside to comment. But please, we are running out of time, so we initiate them quickly. Okay, well, we have to make it to me, but you know, maybe as tomorrow we still have to have your other colleague. Maybe we can spend a few minutes picking up on this topic, if you don't mind. Okay? Yeah, because I think we're running out of time, and I see people getting nervous to go. Okay. So I think okay, but it was a very nice review, and so on. So I would like also your presentations to be a kind of training for making these short talks and so on. And maybe tomorrow or some of the following days, I will give you some hints on how to make an attractive kind of focused presentation when you are one out of the various people to speak, okay? But thank you very much. You made a good job. Thank you very much. Okay, we go. You have to take assignments for tomorrow. So I see. Can you help? It will be just 2 minutes, okay? Don't worry.